Hi everyone, today our topic is evaluation tests. Here we have different types of evaluation tests are available, that are flammability and combustibility. In that, flash point and flame projection we are checked. Next one is physico-chemical characteristics. In that, we have different types of tests, that are vapor pressure, density, moisture content, identification of propellants. Next, performance. In that, we have again different types, that are aerosol valve discharge rate, spray pattern, doses with metered valves, net contents, foam stability, particle size determination, and finally biological testing. In that, therapeutic activity and toxicity studies we will check. Now we will check first one, flammability and combustibility. First one is flash point. So here we are using apparatus, name is tag open cup apparatus. So the product is chilled to minus 25 degree Fahrenheit, and test liquid temperature is allowed to increase slowly, and the temperature at which vapors ignite is called as a flash point. Again I repeated, here we are using apparatus is tag open cup apparatus. The product is chilled to minus 25 degree Fahrenheit, and test liquid temperature is allowed to increase slowly, and the temperature at which vapors ignite is called as flash point. Next, flame projection. Here product is sprayed for 4 seconds into a flame, and the flame is extend. The extent length is measured with a ruler. Here in this video, we will check physico-chemical characteristics. In left hand side given property and right hand side given methods. The first one is vapor pressure. In this vapor pressure, we are doing two methods, that are pressure gauge and can puncturing device. Coming to second property, density. Here we are conducting methods are hydrometer and pinometer. Next, third one is moisture, and here method is Carl Fisher method and gas chromatography. And finally, identification of propellants. We are checking methods are gas chromatography and finally IR spectroscopy. So coming to next one, performance. In performance, we will check aerosol valve discharge rate. Here, the contents of the aerosol product of known weight is discharged for specific period of time. By rewaving the container after the time limit, the change in the weight per time dispensed gives the discharge rate, that is gram per second. Second test is spray pattern. The method is based on the impingement of spray on piece of paper that has been treated with dye talc mixture. The particles that strike the paper cause the dye to go into solution and to be absorbed onto paper, giving a record of spray for comparison purpose. Next test is dosage with metered valves. Reproducibility of doses can be determined by assay techniques, accurate weighing of filled container followed by dispensing of several doses. Containers are then reweighed, and difference in weight divided by number of doses dispensed gives average dose. So next test is net contents. Teared cans that have been placed onto the filling lines are reweighed, and the difference in weight is equal to the net contents. In destructive method, weighing a full container and then dispensing a much of the content is possible. The contents are then weighed. This gives the net content. Next test is foam stability. Here methods involves are visual evolution, time for given mass to penetrate the foam, time for given rod that is inserted into the foam to fall, rotational viscometer. Next test is particle size determination. Here methods are involved: Cassaday impactor, light, scattering, decay. In particle size distribution, methods are involved in two types, that are cascade impactor and light scattering decay. Now we will discuss cascade impactor. The main principle involved in this: stream of particles projected through a series of nozzles and glass slides at high velocity. Larger particles are impacted first on lower velocity stage, and smaller particles are collected at higher velocity stage. Next one, a light scattering decay. Here main principle involved here: as aerosol settles under turbulent conditions, the change in the light intensity of a Tyndall beam is measured. The final one, biological testing. Here two tests is conducted, that is therapeutic activity, second one is toxicity. First one, therapeutic activity. For a inhalation aerosols, dosage of the product is determined and is related to the particle size distribution. For topical aerosols, it is applied to test areas and absorption of therapeutic ingredient is determined. Next, second one is toxicity. For inhalation aerosols, it is exposing test animals to vapors sprayed from aerosol container. For topical aerosols, irritation and chilling effects are determined. This is the last video from the unit 4 pharmaceutical aerosols. Tomorrow we will start unit 5 blood products and plasma substitutes. Thank you.